Good evening, everybody. This is Dr. Patmos. It is February 12th, 2026, and I have what I hope will be an interesting video for you tonight. If you haven't been keeping up with the news, there's been some major developments in the last couple of days. Dr. Vinay Prasad, the director of the Center for Evaluation of Biological Evaluation and Research, or Biologics Evaluation and Research at the FDA, issued a proclamation that basically said that the FDA would not approve Moderna's new flu mRNA based flu vaccine. This was a big deal because they did significant research along the exact lines that the FDA had required of them. And then at the 11th hour, they were denied the application. And it's not because it um, didn't work. It was because Dr. Prasad, by himself, decided that he didn't think the study design was adequate. At the same time, there are rumors, not just rumors, but actual statements from Dr. Malone, who's on the CDC ACIP committee, that um, there will be a recommendation coming forward from the next ACIP meeting that will likely restrict further the use of mRNA vaccines, the mRNA COVID vaccine in particular. Dr. Malone even said that he's not deaf to or blind to or ignoring the calls for mRNA vaccination to be taken off the market entirely. So I thought it would be, given that Dr. Malone is really probably, in my opinion, the most influential and powerful voice in the world when it comes to mRNA vaccine technology, even more than Drs. Carrico and Weissman, who won the Nobel Prize for the messenger RNA vaccine. I think Dr. Malone is really the most powerful voice. And it is really Dr. Malone who I think is behind the groundswell movement against messenger RNA vaccines. And I thought it would be interesting to go back and look exactly in detail at what his role was in the development of the vaccine or in the development of the technology for RNA transfection. Now, as you know, Dr. Malone has put on his CV and stated innumerable times in innumerable places on social media and elsewhere that he is the inventor of the RNA messenger RNA based vaccine. He believes that he invented it. At times, he has also said that he invented the technology behind the vaccine. So there's a little bit of a confusion there. Did he invent the vaccine or did he invent the technology underneath it? Many people dispute this, including the New York Times and others, who say that his description of his role is a significant exaggeration. About 4 years ago, I looked at his paper, his 1989 paper, in which he described the research and showed that he acknowledged Dr. Feldner, his mentor, throughout the paper, and basically got the idea from Dr. Feldner to use lipofection to transfect RNA. So my argument was that he did not invent this by himself, that it was uh, based upon what he had gotten from his mentors, Dr. Feldner and Dr. Verma. But I didn't really go into great detail on exactly what the difference is between Malone's '89 paper and Feldner's 1987 paper, where he first described transfection of DNA. In this article tonight, in this presentation, I did that. I went and in detail compared the materials and methods of the two papers. Now, this is very technical and very dry and probably very boring for most of you. And at the end, I will try to put in a simple analogy in place here that will make it understandable for those of you who are not molecular biologists or biochemists. But basically, in a nutshell, in 1987, Dr. Feldner and his associates, Dr. Malone wasn't one of them, he was still in his master's program at the time, published a paper in which they showed that they used this novel material called lipofectin that Dr. Feldner developed to transfect DNA. In other words, to take DNA and get it to go into cells. That's a big deal because it had never been established before. There was never been a way before to take DNA and push it into cells. And Dr. Feldner described that process. He created a process. He created a material, lipofectin, basically a lipid globule that could, in which you could put the DNA and mix it in with cell cultures, and it would get into the cells. And he published that in 1987. In that article in 1987, he suggested that this same method could be used to transfect RNA and oligonucleotides. So the idea of using lipofection to transfect RNA came from Dr. Feldner and his colleagues in 1987 in an article in the Proceedings of the National Academy of Sciences, which I have attached a link to. 2 years later, Dr. Malone, who had just finished, he was not a doctor at this point, he had just finished his master's degree in biochemistry or biology at UC San Diego, and was in a graduate program, MD, PhD program, and was assigned to the lab of Dr. Inder Verma, and also supervised by Dr. Feldner. And he was tasked with working on the project that was suggested in Dr. Feldner's paper, figuring out how to actually transfect RNA into cells. Well, it turns out that the process that Dr. Feldner developed didn't work very well for RNA. RNA is a different molecule, and that same process didn't work, and it required some changes. It required changes in some of the reagents and some of the incubation time and some of the dilution ratios and measurements and so forth. It required some significant material changes. And so I looked very carefully at the 1989 paper that Dr. Malone wrote. He was not a doctor at the time. The secondary authors were Feldner and Verma, his supervisors. So I compared the materials and methods very diligently between both papers. And although there is a lot of overlap, there is some very significant differences. And it is clear that in the 1989 paper, Dr. Malone had to significantly tweak the process. He had to use some different reagents and a different methodology. Now, the changes he made, he did not invent. Those changes were already out there in other research, but he did a really wonderful job of getting those other changes and recombining them and figuring out a way to make it work for messenger RNA. And that was really a great contribution. As a grad student doing the actual bench work at the laboratory bench with the pipette, Dr. Malone made a very important contribution. He actually changed the recipe to make it work for messenger RNA. He didn't do that in isolation. He was part of a big molecular biology ecosystem that encompassed UC San Diego, Salk Institute, Vical Labs, Dr. Feldner, Dr. Verma, and many other grad students and other researchers. And in that collaborative environment of translational research, using an iterative process, Dr. Malone was able to successfully transfect RNA. Kudos to him. That was a really big deal, but it was not done in isolation. And then there was, a dispute that followed over who got most of the credit and blah, blah, blah. I'm not getting into that dispute. But what I wanted to do was put into perspective what Dr. Malone's role was relative to Dr. Feldner and Dr. Verma. And let me make an analogy to make this simpler for those of you who are not biochemists or molecular biologists. Imagine that you came up with a recipe and some special ingredients to bake a fantastic new chocolate cake, a chocolate cake that had never been baked before with unique ingredients. This is a special chocolate cake, and you came up with it. And you decided to write an article with the recipe. And you said in the article, I think this recipe could be used to make a great red velvet cake. And you hired a junior baker to come into your bakery and tasked that junior baker with the job of making the red velvet cake with your chocolate cake recipe. Well, it turned out that the chocolate cake recipe just doesn't work very well for red velvet cake. So the junior baker had to come up with a tweak of the recipe. He had to use some different ingredients, cook it for a longer period of time, and make a little difference in that recipe. He didn't come up with that by himself. He had help doing that. But he basically implemented a new recipe that was successful in creating the red velvet cake that was suggested by the senior bakers. So the question I have for you, who gets credit for that? Who actually was the first person to bake the red velvet cake? Yes, it was the junior baker. He baked it, but he didn't do it by himself. He did it with help from the senior bakers, and he got the idea from the senior bakers. It wasn't his idea in the first place. So this is all very complicated, and I think it puts into perspective exactly what Dr. Malone's role was. It was an important role. I don't mean to diminish the significance of what he did. He did important research as a student, utilizing the idea that came from Verma and Felgner and the process that came from Verma and Felgner, but he had to tweak that process in a significant way to make it effective. And for that, he deserves a tremendous amount of credit. But I think it is unfair. It's an unfair characterization to say this was done in isolation, that it was all his idea. That's just not what the historical record shows. It wasn't important. These two papers, the 1987 and 1989 paper, were very important because without that research, the development of an RNA-based vaccine would have taken much longer. I'm sure others would have come up with the answer, but this research did move it along faster. And so what we have now is we have messenger RNA COVID vaccine that has been shown to be extraordinarily effective and extremely safe. There is no credible evidence that this vaccine is dangerous. That's all exaggerated and not based upon the evidence. There's clear-cut evidence that the messenger RNA-based COVID vaccine saved millions of lives around the world. Even the congressional committee that investigated COVID concluded that the RNA vaccine was a great success and saved millions of lives. Well, here we are on the cusp of taking it away from, at least in the United States. It will still be used around the world. It just won't be used in the United States. We're losing, in an incremental way, we're losing the messenger RNA-based COVID vaccine. But wait, there's more. We just lost the messenger RNA-based influenza vaccine that was denied by Dr. Prasad. And it's highly likely that we're going to lose the access to the messenger RNA vaccine, COVID vaccine for pregnant women and for others, and probably incrementally, as long as these people are in power, messenger RNA vaccines will be gradually taken away. But it's worse than that because messenger RNA vaccine technology has also already been shown to be effective in the treatment of melanoma and other cancers. And that research is likely to be stalled as well. So the problem we have here is we have someone who has an agenda and a chip on his shoulder and is doing everything he can to slow down the progress and the deployment, and actually reverse the progress and the deployment of messenger RNA-based vaccination. And I think it is important to put into historical context exactly what his contribution was and what it was not. So hopefully you found this interesting, and maybe you feel like having a piece of chocolate cake now. Have a good evening.